An introduction to bioinformatics. The aims for the lecture are to introduce some basic concepts of bioinformatics. First of all, what is it? Then we're going to talk a bit about homology and its importance, how we store data, how we do sequence alignment, data searching in order to identify homologues, and finally talk a bit about annotation, the human factor that's involved in all of this. The objectives for the lecture are that you will be able to define homologues, orthologues and paralogues. You'll be able to give examples of databanks and show the difference between a database and a databank, and between a databank and a search tool. This is a very important distinction, and we'll talk about that later. You'll be able to describe the basic idea of sequence alignment and the differences between global and local sequence alignment. You'll be able to describe the principles of identifying homologues and functional motifs and the key statistics that are involved in deciding whether something is truly likely to be a homologue. And finally, you'll be able to discuss the problems of annotation. First of all then, what is bioinformatics? Well, everyone's definition is different, but the simplest definition is the application of computers to problems in biology. Expanding on that a bit, it's aiding the biologist in creating, storing, searching and analysing biological data, particularly sequences of both proteins and DNA and structures, but also various types of omics data from genomics, proteomics, transcriptomics, metabolomics and so on. Presenting those data in a way that biologists can use and applying the analyses that we do in order to make predictions. To set the scene with the background, biology is now overwhelmed with data. Traditionally, biology was quite a data-poor subject. It involved people going out and drawing pictures of plants or furry animals or whatever and trying to classify them. Now, with things like deep sequencing that allow us to obtain genome sequences really quickly, it's completely overwhelmed with data. The human genome alone is about 3.2 gigabase pairs, that's 3.2 billion base pairs of data. And that's enough raw data to fill about 5 CD-ROMs or 1 DVD. The DNA sequence databanks are now approximately 8,542 billion base pairs of data. Now, it passed 100 billion in August of 2007, although they had also said that in August of 2005, but there was some kind of duplicate counting done then. So I think the August 2007 number is more applicable. But you can see how the amount of data has gone up between 2007 and 2020. So just a few other statistics to watch how it's increasing. In January 2019, so just kind of 18 months ago, it was 3,724 billion base pairs. A year before that, 2,715 billion base pairs, and a year before that, 2,042. The current number is roughly equivalent to having sequenced the human genome 2,669 times. So a huge is a human genome equivalent. Now, some of you might be thinking, well, that's actually rather low, because you may have heard of things like the 10,000 Genomes project, or now the UK 100K project to sequence 100,000 genomes. But in those cases, all we do is store the differences in the genome data between the different genomes, because most of it is pretty similar. So these are kind of unique sequences that we're talking about. That's also equivalent to nearly 2,800 years worth of issues of the New York Times. And that's a really kind of large, thick newspaper, particularly at the weekends. We also have about 169,000 protein structures. So we really have huge amounts of data, and this is before we consider any of the omics data that are available now, and things like specialist resources, for example, the deep sequencing of antibody repertoires. And it keeps growing. So on the basis of those sorts of numbers, DNA data doubles roughly every 18 to 24 months, and the structural data doubles about every 6 years. These are somewhat old graphs, but the trends are exactly the same. So the graph on the left shows the growth of the International Nucleotide Sequence databases, and there are 3 of these. The bluey purpley colour is GenBank, which is the American resource. Yellow is EMBL, which is the European resource, and the red wine colour is DDBJ, which is the DNA Databank of Japan. Now, in fact, these 3 resources all exchange data on a very regular basis, so they end up having the same data in them, but in different formats. The graphs then represent the number of sequences deposited in those particular resources. So obviously people in the US tend to deposit their data with GenBank, in Europe with EMBL, and in Japan in DDBJ. The right-hand graph shows you that the trend applies both to the number of base pairs of DNA and to the number of sequences, and the trends really follow each other very closely. The Protein Databank, which is where we store the structure data, currently consists of about 150,000 structures that have been solved by X-ray crystallography, about 13,000 from NMR, and 6,000 from electron microscopy. And this number in particular is going up very rapidly at the moment, as there have been a lot of advances in electron microscopy techniques for identifying the structure of proteins rather than just looking at lower resolution things. However, these structural data only represent something like around 65,000 sequences if you put in a sequence identity cutoff such that you throw away very, very similar sequences. So you can see there that we're talking about 65,000 sequences for which we know structures. But from the graph on the right here, and this is 15 years ago, remember, we're talking about 54 million sequences in GenBank. As we said, the human genome is about 3.2 gigabase pairs, 3.2 billion base pairs. Now that's 30 times the size of something like C. elegans or Drosophila. But only around 2 to 5%, probably about 3% of that genome is actually coding for proteins. And more than 50% is made up of repeated sequences. Now those repeats can be small tandem repeats, things like AT, 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 or they can be repeats of things like transposons that move around the genome. So things like line elements and sign elements are very typical. But about 35% of genes have alternative splicing. It's estimated that roughly 3% of coding DNA codes for about 20,000 proteins. Now, 35% of those have alternative splicing, which means, of course, that we've got more actual proteins being produced. And it's estimated that, on average, each gene has about 3 splice variants. So that means that we're talking about maybe 50 to 60,000 different proteins that can be produced. So what are we trying to achieve using bioinformatics? Well, it's all really about the central dogma of biology, that DNA sequence encodes protein sequence, which encodes protein structure, which then has a function. Now, going from DNA sequence to protein sequence, if we know the parts of the DNA that encode protein, then that's really quite trivial. But actually identifying the regions of DNA that are coding genes is much more difficult. Going from protein sequence to structure, well, there are various different ways of doing that. We can do things like homology modeling, where you take information from one known protein structure and use that to predict another structure. But people are also attempting to do ab initio predictions, where they're trying to fold up a protein in a computer, but that's still really very difficult at the moment. So from both protein sequence and protein structure, we can get to protein function. Again, in terms of prediction, that's really very difficult, although there has been some success in that area now. But when we understand the detail of protein structure and we understand the function, then we can see how that structure influences the function, and we can look at things like mutations and understand how those damage protein function. Now, once we understand the link between structure and function, we hope to be able to modulate the function. In other words, design drugs. Those drugs can be inhibitors, they can be agonists or antagonists of receptors, so either switching a receptor on or switching a receptor off. And at least in principle, although as far as I know there are no drugs that work this way at the moment, if you have a mutation, you might be able to restore the protein structure and function by binding a drug to that mutant protein. At this point, we're going to take a short break, and I want you to visit the Protein Databank web pages at www.rcsb.org. Take a look at the molecule of the month. Obviously that will change each month, but just scroll down to the section on exploring the structure and you'll be able to view the protein interactively with a web-based structure viewer called JSMol. From the main page at rcsb.org, you can search for a protein, perhaps hemoglobin or myoglobin or something that interests you. You can then view the 3D structure on the web page or download it to view it with free software such as PyMol or Rasmol that you can install on your computer. Now, I've mentioned homology already, but this is a really key concept and one which really drives an awful lot of what we do in bioinformatics. Homologous proteins or genes have descended from a common ancestor. That is the definition of homology. So we have on the right protein 1, protein 2, both share this common ancestor. It's therefore wrong to refer to percentage homology. That is just not possible. You're either homologous or not, just as you can't be 50% pregnant or 20% dead. Now, of course, with pregnancy or death, you could measure how long somebody has been pregnant for or dead for. And in principle, at least, you could go back in time and say, when did these proteins descend from this common ancestor? When did they diverge? Of course, we can't actually go back in time, but we can look at the sequence identity or sequence similarity between the proteins in order to estimate how long ago they diverged. Now, within the concept of homologues, there are two main types. The first is orthologues, or orthology means that we are talking about homologues resulting from a speciation event. So that could be my hemoglobin compared with mouse hemoglobin or rat hemoglobin. Paralogues, on the other hand, result from gene copying within a species. So myoglobin and hemoglobin are homologous proteins, but they are both present in humans and both present in mice and rats and so on. So those are paralogues. As I said, we can look at the percentage sequence identity or percentage similarity between two proteins, and that can be used to infer whether they are homologous or not. So sequences of at least 100 amino acids in length with at least 35% sequence identity are always homologues. You don't get that level of similarity or identity between proteins unless they've descended from a common ancestor. But sequences with much lower sequence identity may be homologues. And sequence similarity may be a more useful measure than identity. And I'll explain a bit more about what I mean by similarity in a few minutes. In fact, sequence identity between homologues can be very low. You can find instances of proteins that you absolutely know are not homologues because they share no structural similarity, no functional similarity, and they can have 15% sequence identity. But proteins that we know are homologues because they have a very, very similar structure and they share some functional similarity can have sequence identities as low as 10%. Why then is homology so useful? Well, I've already mentioned some of the characteristics of homologues. Because they have derived from a common ancestor, some of these characteristics will be similar. They will have a related function and a related structure, and that means that we can infer functional information and structural information from known information about one protein. We can infer that onto another protein. Let's take the example again of hemoglobin and myoglobin. I've rendered those in different ways. I'm using a kind of ribbon representation here, and here I'm just using a backbone worms representation as it's known. And the reason that I've done the two different representations is really just so that when we overlay them on the right, you can actually see some difference between them. So you can see that the structures really are incredibly similar. There's a little bit of a difference up here on the top right and a little bit of a difference up here on the top left, but really the core of the proteins is almost identical. Now these two only have 27.7% sequence identity, so that's really quite low. It's certainly below that threshold of 35% that I mentioned earlier. But as well as having a very similar structure, they also have a very clearly related function in terms of oxygen binding. Of course, hemoglobin is involved in transport and myoglobin is involved in storage. However, this is a good illustration of the fact that if we can identify homology, then we can do transfer of annotations in terms of function, and we can do transfer of structural information. And that allows us to do what's known as protein homology modelling, or protein comparative modelling. So that means, why use one of these? The top picture here shows you a small lab-based X-ray crystallography machine, and the bottom picture shows you an NMR machine. Why use this sort of expensive, large, difficult to maintain equipment if you can just use a desktop PC, or indeed a laptop? As I said earlier, bioinformatics and biology in general is now overwhelmed with data. So a very important aspect of bioinformatics is how we store those data. Now, I like to make this distinction between a database and a data bank. It's not a set in stone distinction. Other people may define these things slightly differently, but I think this is a useful way to look at it, and it really mirrors the way that the terms tend to be used in the literature. A data bank then is simply a collection of data, normally in simple text files, the sort of thing that you could view with Notepad or Word or something like that. And that collection of data has no fixed associated query tool. That means you can download all the raw data and you can handle it however you wish. You can use people's search tools that they have developed. You can use your own search tools that you decide to write. On the other hand, a database is a structured collection of data, and it's hidden behind a fixed search tool that you cannot change. Now, that search tool might be what's known as a relational database, and this is a piece of software that allows you to store data in tables and search those data. Probably the best known example is Microsoft Access, which is kind of the extra member of the Microsoft Office suite. But we can also think of a website as being a database. If the only way of accessing the data that we're interested in is via a website, then we could define that also as a database. Now, the vast majority of bioinformatics resources are in data banks. That means, as I said, that they are plain text files that you can download and process as you wish. Now, for a lot of those, there is also an associated database that typically will be a website that allows you to access the data. But the fact that you can download it and have it all locally means that it will be classified as a data bank. Now, we can divide data banks into different types. A primary data bank contains raw data. So this might be sequence data, structure data, and so on. And there may be some detailed annotations associated with that. On the other hand, a secondary data bank contains data that are derived from information in primary data banks. For example, we can divide proteins into homologous families, as we have just said. And those homologous families are likely to have common characteristics, such as what are known as sequence profiles, characteristics of the whole sequence, or patterns of amino acids that are conserved because they're related to the protein folding or the protein function. Secondary databanks are almost always highly annotated with information about these families. Let's look at some examples of primary databanks. Now, we've already encountered GenBank, EMBL and DDBJ. EMBL is now called EMBL ENA, so that's European Molecular Biology Lab, European Nucleotide Archive. These three resources, as I mentioned before, contain DNA sequence data. Now, they may also contain translations of the regions that encode proteins, but their primary function is to record the DNA data. The second line of examples here are protein sequence databanks. So we used to have a situation where we had in Europe what was known as Tremble, which was the translation of the EMBL ENA databank, and SwissProt, which was the more carefully annotated, defined sequences of proteins, which didn't just come from automatic translation. And in America, the equivalent resource to SwissProt was the PIR, the Protein Identification Resource. Now, some years ago, these resources, PIR and the people who looked after SwissProt and Tremble, they merged together to form UniProt KB, which is Unified Protein Knowledge Base. And we still have SwissProt and Tremble within that, but this resource is the key place where we store protein sequence data. Now, these resources, as I said, they contain basic sequence data of DNA or protein. They may also have feature information, things in DNA like splice sites, signal sequences in protein, location of disulfides, active sites, and so on. The DNA databanks, as I mentioned, may also contain translations to protein, and those could be known translations or predicted. Now, that point about prediction, as I said in my overview of what we try to do with bioinformatics, that prediction of which regions of DNA encode protein is not trivial. And given that these gene prediction methods are imperfect, then you'll find that in a resource like SwissProt, a protein that has been identified from genome data without being verified by experiment will be labelled as hypothetical. A couple of other examples of primary databanks. The PDB is the protein databank where the structural data are stored. The enzyme databank stores enzyme classifications, so-called EC numbers, which is a kind of hierarchical classification of enzymes based on what they catalyse. Moving on then to secondary databanks. Here are a few examples: ProCite, Prints, Blocks and Interpro. And as I said before, these contain secondary or derived information. They have patterns typically that characterise a particular protein family and almost always have detailed annotations. Now, Interpro is a kind of meta-resource that contains the information from these other resources. We'll see in a few minutes why these sorts of things are useful, remembering that they characterise patterns that occur within protein families. You'll see why that is important in just a minute. Now, sequence alignment is really a key tool in bioinformatics, because from sequence alignment we can calculate sequence identities and sequence similarities. So what do I mean by an alignment? Well, here are two short pieces of sequence, and this is one possible alignment. I've aligned the leucines, serines, aspartic acids, leucines here. These don't match, and I've inserted a gap into this sequence. Alternatively, I could mean something like this, where I have aligned the N-terminal part and put a gap at this end. Or I could even mean something like this, where I have aligned the S's and L's and that's all. The rest really doesn't match anything. However, what we normally mean by a sequence alignment is inserting gaps into one of the sequences or both of the sequences such that we optimize the total number of identities or similarities between amino acids. Now, in this particular case, we need to insert a gap around here but it's not obvious whether this leucine should be aligned with this first one or with this second one, as I've done in the example over on the right. Now, just by looking at two sequences, there is really no way to identify which of those is the correct alignment. And what do I mean by correct? Well, largely it tends to mean what maps to the structure. So if we had a structure of this bottom bit of sequence, would the two residues that are inserted in the top sequence tend to go here or here? Now, having more sequences than this, so if we had a multiple sequence alignment of say 10 or 20 sequences, we might get a bit more of a signal about where the insertion or deletion actually tends to occur. And of course, if we actually had a structure of one of these, it would be more easy to see where an insertion or deletion would be likely to be accommodated. And of course, if we had both structures, then we would know completely where the insertion or deletion occurs. How then can we examine the alignment between two sequences? Well, if I take the same simple example that I showed you on the last slide and plot one sequence along the top and one sequence down the left-hand side, then anywhere that the two sequences match, I can put a red blob. So two glycines here, serines here, but this serine, there's also a match with a serine there. Now, by looking at that, you should be able to see that there is a kind of vague diagonal line down here, but it's broken at this point in the middle, and that's where the insertion occurs in this sequence compared with this sequence. Now, the fact that we have two blobs on the same line indicates our ambiguity about whether this leucine should align with this one or with this one. Now, in practice, of course, these sorts of plots can become very noisy. So what I have here is the light chain of two different antibodies, and you can see on the left, again, you should be able to see that kind of faint diagonal here wherever there are matches, but we can do some noise reduction, which means in this case that we have only put a dot if 3 of the residues in a line of 7 match. So we're taking a little window, and wherever we have 3 out of 7 matches, we put a dot in this version. So now you can see much more clearly this diagonal line coming down here, and of course there are little off diagonals as well, where there are small motifs within the sequence which show similarity to one another. Now, I've talked a fair bit about both sequence identity and sequence similarity without really explaining what I meant by sequence similarity. The easiest way of scoring one sequence against another is to use identity. So if we have a serine in one sequence, we need to have a serine in the other sequence for it to count in terms of scoring the identity between two sequences. So it's 1 for a match, 0 for a mismatch. But we can also use more complex scoring schemes which account for the similarity between individual amino acids. So for example, serine and threonine are really very similar to one another. Threonine just has an extra CH3 group attached on the side, whereas serine and tryptophan are really very different. Tryptophan has a double ring structure, serine just has an OH group. On that basis, we can create some sort of matrix to score the similarity between any pair of amino acids. And this is typically derived from analysis of aligned homologous proteins, and we see what substitutions are observed. And that's telling you over evolutionary time how one amino acid is able to substitute for another. You can also produce scoring matrices based on things like physicochemical characteristics, but the ones based on evolution tend to be the ones that are most widely used. Now, this was first done by Margaret Dayhoff in 1978, and that produced what's known as the Dayhoff mutation matrix, or PAM matrix. This type of thing was improved by Henikoff and Henikoff in 1992, and they derived the so-called Blossom matrices. And this is a family of matrices such as Blossom 80, Blossom 65 and so on, which are designed for homology searching at different sorts of similarity levels. So if you're looking for proteins that are very similar, you use a high scored Blossom matrix. If you're using this to look for sequences that are quite different from one another, you use one of the lower matrices like a Blossom 35. Now this is the Dayhoff matrix, and you can see here that we have scores ranging from some negative numbers like -4, -6 and so on, up to positive numbers like 12 in this case for cysteine, converting to, in other words, being conserved as a cysteine. Scores of 0 mean that a particular change happens at the same rate that you would expect by chance. Scores less than 0 represent changes that are observed less frequently than you'd expect by chance, and scores above 0 are ones that you observe more frequently than you expect by chance. Now, if we take the example that I gave before of serine versus threonine, if we find serine along here and threonine here, it's a positive score. It's only one, but most of the positive scores that are not on the diagonal tend to be fairly small. Now, serine and threonine, as I said, are very similar to each other. They share very similar characteristics. Another good example is tyrosine and phenylalanine, which both have a phenyl ring. Tyrosine just has a hydroxyl group added onto the end. So tyrosine is Y, phenylalanine is F. So those score very highly. There is nothing else that really looks like them in terms of their physical characteristics. Similarly, I mentioned up here isoleucine, leucine and valine. So if we look for isoleucine here versus leucine, that scores 2, and versus valine, that's at the end here, that scores 4, and valine versus leucine scores 2. So you see they're all positive scores. Now if we take something like tryptophan and serine, which obviously are very different, tryptophan, as I mentioned before, has this double ring system, whereas serine just has a hydroxyl group, you'll find there that you have a score of -2. So given a scoring matrix like this, we can calculate what the maximum score you could get for one sequence scored against itself or the other sequence scored against itself. Now, comparing the two sequences, obviously you can never get more than the lower of those two. So if you score one sequence against the other, you can then calculate a similarity based on the observed score versus the maximum score you could obtain, and that gives you a percentage similarity. Okay, at this point we're going to take a short break and have 3 little tasks for you. The first of those is to draw your own dot plot to compare the two following sequences. Second, if we assume that the alignment doesn't require any insertions and deletions, in other words it appears exactly as shown here, calculate the sequence identity for those two sequences. And thirdly, using the scoring matrix, the Dayhoff matrix that I showed you on the previous slide, calculate the sequence similarity for these sequences. Remembering that what you need to do is sum up the scores for the alignment and then sum up the scores for the first sequence aligned against itself and the second sequence aligned against itself. And then the sequence similarity is the score for sequence 1 aligned with sequence 2 divided by the minimum of the scores you got from the two self-alignments. Okay, so when you drew your dot plot, you should have ended up with something that looks a bit like this. So we have the two sequences, one along the top, one along the side, and everywhere we have a cross, we have a match of the amino acids. I've highlighted then in red the ones that really kind of fall down the diagonal. As you can see, the ends really don't match at all, but you can see from this that these do fall on the diagonal. There are no insertions or deletions required in order to align one sequence with the other. So in calculating the sequence identity, you should have put the two sequences and found the amino acids that match, and you should have found that 6 out of 12 of the amino acids match, giving you 50% sequence identity. On the other hand, in calculating the sequence similarity, this is the comparison of the two sequences, looking up their scores from the Dayhoff matrix. We add those up to produce a score of 33. If we score the first sequence against itself, we obtain a score of 56. The second sequence against itself, we obtain a score of 73. And that means that we're going to divide by this lower score, because you could never get a score greater than this if you're using this sequence. Therefore, our percentage sequence similarity is going to be 33 divided by 56, which gives us a similarity of 58.9%, which, as you see, is higher than the identity, which was just 50%. That's all very well, but if you're talking about aligning lots of pairs of long sequences, you don't want to be doing that by hand. And so that's where an algorithm called dynamic programming comes into play. So dynamic programming, or DP, is applied to the scored dot plot, which can be scored by similarity or identity, to find the optimum path through the matrix and hence the best alignment between the sequences. Now, this was introduced in 1970 by Needleman and Wunsch, and actually Needleman and Wunsch were two medics, and they published this seminal paper in the Journal of Molecular Biology, which is probably one of the most cited papers ever, certainly in the bioinformatics field. And then they returned to trying to cure the common cold or whatever, and never published anything again. The algorithm that they came up with, they didn't call it dynamic programming, but it turned out that this was actually an algorithm that had been around since the 1950s and is used quite widely in computer science for solving what's known as the travelling salesman problem. So if you imagine you have a travelling salesman who has to move between a set of different cities, then what is the optimum path between those cities when you take into account traffic speed and distance? And Smith and Waterman in 1981 realised that what Needleman and Wunsch had done was actually a form of dynamic programming, and so they formalised the work that had been done by these two medics, and they then modified it somewhat, because what Needleman and Wunsch had done was what's known as global sequence alignment, whereas Smith and Waterman modified the algorithm to produce what's known as local sequence alignment. So global sequence alignment is, as the name suggests, the idea of taking the whole of one sequence and trying to align it with the whole of another sequence. So we end up with gaps, in this case 2 in the top sequence and 1 in the bottom sequence, and this little segment aligns with this segment, that aligns with that, and so on. The modification that Smith and Waterman made to produce local alignment means that we have essentially one subsection of one sequence which matches a subsection of another. So in this case, this little segment of sequence here matches to here, then there's a gap, this then matches to here, this has a gap in it, that bit matches with that bit, and so on. Why is that important? Well, the way that proteins evolve really depends on the concept of domains. Now, there are differing definitions of a domain, but you can think of a domain as an independently folding unit of a protein. But you can also think of it as a unit of inheritance. This means that you can mix and match domains during evolution such that you can build up proteins with different functions. So this is a kind of cartoon representation of a number of different proteins which have common domains indicated by the colours. So the red ones are adenine nucleotide alpha domains, and you can see these are found in all of these different proteins, but paired with different things. So in this case it's an N-terminal nucleophile domain. In this case it's a molybdenum cofactor binding domain. And this means you can think of domains as like Lego blocks that do different functions. For example, you might have something that binds and breaks down ATP to release energy, and you could think of that as being like a battery for a protein. Now that means if you tried to align sequence 5 with sequence 2 using global alignment, it really wouldn't work very well. Let's take an extreme example where we have domain A followed by domain B in one protein and domain B followed by domain A in another protein. Now, a global alignment between those two would really make no sense. You can't align A with B or B with A. They are not related domains. A local alignment, on the other hand, will do what you would expect to happen. It will do an alignment of A with A or of B with B, depending on the parameters you choose and the order in which the alignment's done. Let's have a look at a real example of that. It's an artificial example, but it's looking at real sequence. So here we have sequence 1 consists of this domain followed by that domain, and sequence 2 of that domain followed by this domain. If we try to do a global alignment between them, this is the alignment we end up with. And this gives us 48.39% sequence identity with no tails. And what I mean by no tails is that where there are insertions and deletions that occur just at the end of the sequence, I don't include those in what I divide by. So I'm just taking the number of matches that we find here divided by the length of this region between the two vertical bars. If we did a local sequence alignment with the same pair of sequences, this is what we end up with, and we get 100% sequence identity with no tails. So again, ignoring these regions here. This is the output from a program of mine called NW, which does the Needleman and Wunsch sequence alignment. This is the total score for aligning this sequence with this sequence, and here I've normalized it by the length of the overall alignment. This is the similarity score that we described before, so that's taking this score of 73 and dividing it by the lower of the two self-alignment scores. Now, when we come to describe the identity, where we're just scoring the number of matches divided by something, the question is, what should that something be? We could divide by the overall length of the alignment from here to here. That would give us this value. We could score over the alignment length with no tails, which is what I mentioned on the previous slide. So from here to here. We could divide by the length of the shorter sequence, and that's quite commonly done, because again, you can never score more than what the shorter sequence has. You can score over the aligned residues. In other words, you ignore these ones where there's a gap in one of the sequences in calculating the length of the alignment. You can score over just one arbitrary sequence, and that's quite useful when you're doing things like protein modeling, because you want to know what percentage of residues in your target protein that you wish to model are represented by residues in the parent that you're basing the model on. So there is this question of what you divide by, and as I show here, the most common thing is to divide by the length of the shorter sequence, or perhaps a sequence of interest, and then the other common thing is the sequence similarity score. As I also mentioned briefly earlier, this concept of sequence alignment can be extended to multiple sequences to produce what's known as a multiple sequence alignment, or an MSA. Now that allows us to find conserved sites. So for example, all these positions indicated with a star are completely conserved across all of the sequences that we're looking at here. The ones that have a colon are very similar amino acids across all of the sequences. The ones with a dot are somewhat less similar, and the positions with no symbol at the bottom are residues that are quite different. Now, we can also use multiple sequence alignment to generate phylogenetic trees, and this is the basis of how this sort of thing is done. Let's move on now to identifying homologues. The method that's used for doing identification of homologues depends very much on how distant the homologue is that we're trying to find. So when we're looking for things with fairly high sequence identity, automatic pairwise type approaches of the sort that I've mentioned with just doing sequence alignment are perfectly fine. When we get to somewhat lower sequence identities, using a consensus approach is more sensitive. Now, I briefly mentioned the fact that we can extract key residues from a multiple sequence alignment, and these can be used to generate patterns which are then stored in things like secondary databanks. So that's the kind of consensus approach. We'll come back to that in just a second. And then we can also generate what's known as a profile, which again I'll come back to in a moment, which allows us to look for more distant homologues. So here we're talking about below 20% sequence identity. And as I mentioned earlier, we can have examples where sequences which are clearly not homologues can have 15 or 20% sequence identity, whereas things that are homologues can have 10% sequence identity or less. So at these very low sequence identities, we can use things like structure prediction, which allow us to predict certainly the secondary structures quite accurately, and then use techniques like fold recognition to see if we can match the series of secondary structure elements or the sequence itself onto some sort of library of different protein folds, and use that as a way of identifying homologues. In practice, using dynamic programming of the type that I've described already in order to search these huge databanks that we now have of sequence data is far too slow. And that's led to the development of specialist software that allows us to search these very large databanks much more quickly. And there are two key approaches. One is known as FASTA and one is known as BLAST. And the details of the way that these are implemented is rather different, but they both use the same general approach, which is known as a heuristic. A heuristic is a method that will not necessarily give the very best answer, but it will be guaranteed to give you a very good answer in a shorter amount of time. And both of these approaches are based on some sort of index. So what happens with BLAST is that we index the database by finding locations of short words, and then we look up words from the probe sequence against the index in the database, look for multiple matches, and then extend them to find likely hits and a full alignment. So if you think of having a book, but in that book the index, rather than just indexing keywords, indexes every word that's in the book. Now if you wanted to find the page that had a particular sentence on it, you could look in the index for each of the words in the sentence and find the page number that has the most hits, and that would take you straight to that page. You could then read that page properly to find the sentence in that page. Now it wouldn't matter if there were some errors, that you had got one or two of the words wrong in the sentence. It would still take you to the page that had the most matches, and then your reading is a bit like the full sequence alignment at the end, which is just done on that page. So when you're searching a big data bank, you need some sort of statistic to tell you whether the hits you're finding are likely to be significant. Now, suppose we had a single die and we threw that die. What would be the probability of throwing a 6? Well, clearly it would be 1 in 6. Now what would be the p-value, the probability of throwing 5 or more? Well, there are two ways that you can throw 5 or more. You could throw a 5 or a 6. So it's going to be 2 in 6 or 1 in 3. Now, if you throw the die 12 times, how many times do you expect to obtain a score of 5 or more? Well, we know that the probability for each throw of scoring 5 or more is a third. So we multiply that by the number of throws. A third times 12 gives you 4. You would expect to throw a 5 or a 6 4 times out of 12. This is known as the expectation value. It's simply the probability, the p-value, multiplied by the number of throws of the die. In terms of a sequence search, what you have is a given score for a match between your probe sequence and a hit in the database. You can convert the score into a p-value, which is the probability of seeing this score or better simply by chance, and then you multiply by the number of throws of the dice. In other words, the number of sequences in the databank. So the e-value, the expectation value then, is the number of times you expect to see this score or better purely by chance in this databank or database. Typically then we choose a value of 0.01 or 0.02 or less as being indicative that this is really a homologue. But sometimes we can find homologues which have e-values of 10, which are genuine homologues, yet the sequence searching really gives you no statistical support for them being homologues. However, if you are looking for reliable homologues, using an expectation value, an e-value, is a much more reliable way of knowing that what you're seeing is a genuine match than using something like sequence identity or sequence similarity alone. Now, when I showed you the different options for doing searching to identify homologues depending on the sequence identity, I mentioned consensus patterns and profiles. So the ProCite secondary databank is a databank of consensus patterns. Now, those fall into two groups. Some of them are functional sites, which can be used for giving us information about protein function and things like post-translational modifications. So, for example, the square brackets here mean OR. So serine or threonine, followed by anything, followed by arginine or lysine, is characteristic of a protein kinase C phosphorylation site. So if you saw this in a protein, you would think this is a protein probably involved in something like signaling that is modified by protein kinases. This second pattern, the curly brackets mean not. So this is asparagine, not proline, serine or threonine, not proline. And this is characteristic of an N-linked glycosylation site. So a sugar group is added onto the asparagine. Now, these sites are not sufficient to tell you this is definitely an N-linked glycosylation site, but every N-linked glycosylation site will have this pattern. Now, the second type of pattern that the ProCite databank contains is this type, which is characteristic of a particular homologous family. So in this case we have phenylalanine or tyrosine followed by cysteine, arginine or histidine followed by asparagine or serine, and the round brackets indicate a range. So this means 7 to 8 of anything, followed by tryptophan or tyrosine, followed by cysteine. And this is a characteristic pattern of a homologous family known as Kringle domains. This kind of pattern allows you to identify a Kringle domain even though there may be an awful lot of variation elsewhere in the sequence, which makes it difficult to search and identify just using sequence identity or sequence similarity or the e-values that are derived from those. Now, I also mentioned that using multiple alignment, we can pull out characteristics of a sequence family. So what we're trying to do there is pull out information about functional residues that are conserved in a sea of variability. So looking at that multiple alignment, it's quite hard to see what's going on there, but a profile is a way of seeking out conserved residues automatically. And once we highlight then the conserved regions, you can see that there are a fairly small number of amino acids that are at least relatively conserved throughout all these residues. And what a profile does is to take the 20 by 20 scoring matrix and convert it into an n by 20 scoring matrix. So basically it's saying we don't particularly care what amino acid is at this position, or this one, and so on. But here we really boost the score for histidine versus histidine and lower it for anything else. And these sorts of profiles, which are implemented in programs like SciBlast rather than the normal BLAST, allow us to find much more distant homologues. For the last section of this lecture, we're going to look at annotation, which is the human factor in all this. What do I mean by annotation? Well, references to papers that describe a sequence, for example, the methods that have been used, cross-links between different databases and databanks, feature tables, so indications of segments of a sequence or of a structure that are of biological significance. So that might be in DNA, it might be coding regions, in a protein, it might be an active site of an enzyme. We also often have more general descriptions. The problem with those is that while they're great for a biologist to read, they're much more difficult for a computer to understand and get information out of. So in some cases we may use what's known as a restricted dictionary, which is just a set of particular keywords that we might be looking for, or what's known as an ontology, which you can think of as like a tree dictionary. So for example, the word in question might be human, and that would have a set of characteristics that are specifically human, but it would also inherit all the characteristics associated with the word mammal, and so on. We might also talk about the authors, the list of authors, as being an annotation. Now, in terms of quality, the quality of raw data is as good as the methods that produce it, and these days things like peptide sequencing has largely been replaced by DNA sequencing, and DNA sequencing is now completely automated and rapid and of very high quality. But the quality of annotations is only as good as the people who curate those annotations. Now, in the pre-genome world, if anybody bothered to sequence the DNA of a particular gene, then they were almost certainly a single research group with a particular interest in that gene. And that meant that annotations were grounded in experiments and written by real experts. These days, however, with the huge amounts of sequence data that are available, the huge amount of rapid, cheap sequencing that's done, there's rarely experimental confirmation of even the expression of putative genes or the characterization of gene products. Instead, annotations are often based on the sorts of computer analyses that I've talked about in order to find homologues, or indeed to predict where coding regions of DNA occur. And this has been described as the weakest link of genomics. Getting it right is both labour-intensive and underfunded. And some people have gone as far as to say that this poor annotation really negates the benefit of having genome sequence data at all. Here are a few interesting quotes about the reliability of data in GenBank. Michael Ashburner, he said GenBank is full of mistakes. Douglas Crawford from the University of Missouri said the utility of GenBank has declined greatly. While Terry Atwood, who used to be at UCL, said, "We have to educate people about databases so that researchers don't assume that they are right." Now these are somewhat old slides, but they indicate some of the problems that we can get. So in this first case, there is one gene and one protein product, but 10 different names for it. And Keith Yamamoto from UCSF has said scientists would rather share each other's underwear than use each other's nomenclature. Even worse, in this second case, we have two completely different proteins, MRF1 in yeast and RF1M in yeast, but one of the gene names used for them is MRF1 in both cases. And obviously that can cause major problems. The other thing is that as the genomic data grow and methods improve, we hope that the methods for performing annotation and things like finding the coding regions of DNA will improve. But of course, if annotations improve, then by definition they're also in a state of constant change. And does that mean that completed research projects based on those annotations and those sequences will have to be revisited? Arthur Lesk, in his 2002 book on bioinformatics, said, "Databanks will become a seething broth of information growing in size and maturing, we must hope, in quality." Just to summarise things then that you need to know. We've talked about databanks versus databases, we've talked about primary and secondary databanks, and we've given examples of those. We talked about the definition of homology, orthology and paralogy and why these concepts are important. And we talked about databanks and the fact that these are just collections of data, and therefore you can use a search tool of your choice, such as BLAST or FASTA, or we might just be looking for text matches rather than sequence matches. We talked about the differences between global and local sequence alignments and some of the basic ideas around sequence searching using things like BLAST and FASTA, and the meaning of the E-value. If you get nothing else at all from this lecture, understanding what an E-value means is the most important thing. And finally, we talked about the problems of annotation and the fact that you can't necessarily trust the information in a database or databank.